In the earlier parts of cancer genetics, we had seen the role of proto-oncogenes and anti-oncogenes in the genesis of cancer. Proto-oncogenes encode proteins that promote cell division, and anti-oncogenes encode proteins that inhibit cell division. But abnormalities in proto-oncogenes can cause carcinogenesis. Abnormalities in anti-oncogenes can also cause carcinogenesis. So, mutations in DNA play a key role in genesis of cancer. We can say that cancer arises from abnormalities in DNA, mutations in DNA. But a mutation in a single proto-oncogene is not enough to cause cancer. Similarly, a mutation in a single anti-oncogene is not enough to cause cancer. Cancer occurs from a series of mutations. Actually, this genesis of cancer or oncogenesis is a multi-step process. A series of steps convert a normal cell into a cancer cell, and generally this malignant transformation requires a series of mutations. And these mutations generally involve proto-oncogenes as well as anti-oncogenes. So for oncogenesis to occur, there must be mutations in proto-oncogenes and there must be mutations in anti-oncogenes also. Besides proto-oncogenes and anti-oncogenes, abnormalities or mutations in some other genes also contribute to oncogenesis. These include genes regulating the cell cycle, the genes regulating apoptosis, and the genes that encode the DNA repair proteins. So, mutations in a number of different genes are required to convert a normal cell into a cancer cell, and moreover, all these mutations must occur in the same cell. So if a mutation in a proto-oncogene occurs in liver cell, a mutation in an anti-oncogene occurs in a kidney cell, and a mutation in a DNA repair protein occurs in a skin cell, this is not going to cause oncogenesis. Oncogenesis will occur only when the combination of mutations that is required to convert a cell into a cancer cell, when this combination occurs in the same cell. So the process of carcinogenesis begins with DNA damage. But when DNA is damaged, we have got DNA repair systems that can repair the damage. But if DNA damage is followed by failure to repair damage, that is, the damage is not repaired. If a cell has got damaged DNA, normally this cell should be destroyed. It should undergo apoptosis. So, another abnormality that causes carcinogenesis is failure to destroy cells with damaged DNA. And then there must be mutations in proto-oncogenes as well as anti-oncogenes. So all these steps occurring in a sequence lead to carcinogenesis or malignant transformation. As we grow older, the number of mutations that we have increases. We accumulate mutations with advancing age. And if the combination of mutations that is required to convert a normal cell into a cancer cell occurs in some cell and in the same cell, it will cause cancer. Accumulation of mutations is accelerated if we are exposed to DNA damaging agents. Continuous exposure to mutagenic chemicals, ionizing radiations, and viral infections increases the rate of mutations and we accumulate mutations. Moreover, accumulation of mutations is also accelerated if the DNA repair systems are defective. We have got a number of DNA repair systems, but if any of these systems is defective, accumulation of mutations will increase. Sometimes genetic defects are present in DNA repair systems, and these defects increase the development of cancer. A high incidence of cancers at a relatively young age is seen when the DNA repair systems are defective from birth. For example, nucleotide excision repair system is sometimes genetically defective, and this leads to xeroderma pigmentosum. Multiple skin cancers occur at a relatively young age in xeroderma pigmentosum. The mismatch repair system may also be defective, and this leads to development of hereditary non-polyposis colon cancer. So, malignant transformation requires a combination of mutations in different genes in the same cell. That means initially it is one cell that transforms into a cancer cell, and then this cell divides into two daughter cells, two cells divide into four cells, and so on, and there is a rapid increase in the number of cancer cells. But all these cells have identical DNA, and therefore they are exactly identical with each other. They are clones of each other. So a tumor or a cancer is formed by clonal expansion of a single cancer cell. Initially only one cell is converted into a cancer cell, and then a number of daughter cells are formed which are identical with each other and which are clones of each other. So, for example, this is the cell that initially transformed into a cancer cell, and then from one cell two cells are formed, from two cells four cells are formed, and ultimately a large tumor or cancer is formed having identical cells. When a normal cell changes into a cancer cell, some unique features develop in cancer cells which are not present in normal cells. One such feature is immortality. The cancer cells are immortal cells. They never die. They go on dividing forever. Whereas the normal cells, all the somatic cells, are mortal cells. Now, during embryonic life and fetal life, all the cells possess telomerase activity, and this telomerase prevents shortening of telomeres. But when we are born, all the somatic cells lose telomerase activity, and therefore every time a cell divides there is some shortening of telomeres. And when the length of the telomere decreases below a certain critical limit, the cell stops dividing, it dies. But telomerase activity returns in cancer cells. When a normal cell transforms into a cancer cell, telomerase activity returns. This prevents shortening of telomeres, and therefore cancer cells go on dividing forever. A second unique feature of the cancer cells is that they do not depend upon growth signals for their growth. Normal cells require mitogenic signals, generally in the form of growth factors. But cancer cells do not require growth factors for their growth. They are self-sufficient. A third property is that they are not sensitive to growth inhibitory signals. The anti-oncogenes encode growth inhibiting proteins, but cancer cells are not sensitive to these growth inhibiting proteins. Another feature is that cancer cells are able to evade apoptosis, programmed cell death. So this also prevents death of the cancer cells. Another property of cancer cells is their ability to escape or evade an immune attack. Cancer cells are abnormal cells. They differ from normal cells in biochemical and metabolic features. So the immune system can consider them as non-self cells, that is foreign cells, and the immune system attacks all the non-self cells. But the cancer cells develop mechanisms by which they can evade an attack by the immune system. And then, as we have seen earlier, the cancer cells have the ability to invade neighboring tissues and also to spread to distant sites, which we call as metastasis. And lastly, a property of cancers is angiogenesis. New blood vessels grow into tumors or cancers in order to supply more nutrients to the cancer cells. When the cell changes into a malignant cell or a cancer cell, the metabolic profile of the cell also changes. And the changes which occur in the metabolism of the cell are meant to make sure that the cell grows rapidly. For rapid growth, the cell requires more nutrients. So cancer cells appropriate nutrients. They deprive the other cells of nutrients and they use these nutrients for their own growth. The cancer cells require more energy. So glycolysis is increased to provide more energy to the cells. For rapid cell division, these cells require nucleotides, they require nucleic acids. So synthesis of nucleotides is increased, synthesis of DNA is increased, synthesis of RNA is increased in cancer cells. Synthesis of many proteins is also increased. And since the amino acids are used by the cancer cells for the synthesis of proteins, the other cells are deprived of amino acids. Moreover, the cancer cells start synthesizing certain proteins which are normally synthesized only in embryonic life and fetal life when the cells are undifferentiated. So these proteins begin to be synthesized again when a normal cell changes into a cancer cell. Some of these proteins escape into circulation. They can be detected in circulation. They can be measured. And some of these proteins are useful in the diagnosis of cancer. If we detect these proteins, if we measure the concentration of these proteins in plasma, it helps in the diagnosis of cancer. So tumor markers or cancer markers are compounds, mainly proteins, which are synthesized specifically by cancer cells which leak into circulation, and by detecting these compounds and by measuring the concentration of these compounds in plasma, we can diagnose cancer. And these compounds are known as tumor markers or cancer markers. The commonly used tumor markers include carcinoembryonic antigen or CEA, alpha-1 fetoprotein or AFP, prostate-specific antigen or PSA, human chorionic gonadotropin or HCG, carbohydrate antigen 15.3. Now, carbohydrate antigen is abbreviated as CA, and we have got a number of carbohydrate antigens, but they are not actually carbohydrates, they are glycoproteins. So CA 15.3 is one, another one is CA 19.9, then we have CA 27.29 and CA 125. Plasma CEA, carcinoembryonic antigen, is raised in colorectal cancer, carcinoma of colon and rectum. Alpha-fetoprotein or AFP is raised in plasma in hepatocellular carcinoma. Prostate-specific antigen or PSA is increased in cancer of the prostate. HCG is increased mainly in choriocarcinoma, but it is also increased in testicular cancer. CA 15.3 is increased in plasma in breast cancer. CA 19.9 is increased in pancreatic cancer. CA 27.29 is also raised in cancer of the breasts. CA 125 is increased in ovarian cancer. So these are the tumor markers or cancer markers which are usually measured in plasma to help or aid in the diagnosis of cancer. But a rise in the concentration of tumor markers in plasma does not occur early in the disease. It occurs when the disease is fairly advanced. But the success of treatment of cancer depends upon early diagnosis. It is only when the disease can be diagnosed early and treated early that we can get rid of cancer. So, these cancer markers are not useful in early diagnosis of cancer, but they are useful in monitoring the response to treatment. When we treat cancer by surgery or by chemotherapy or by radiotherapy, and we find that the concentration of cancer markers is decreasing, it means the patient is responding to the treatment. So, response to treatment can be monitored by serial measurement of cancer markers in plasma. And moreover, these cancer markers may be helpful in detection of recurrence. When we treat cancer by different means, all the cancer cells may not be destroyed. Some cancer cells may remain. And if only a few cancer cells remain in the body, they can multiply again, and this can result in recurrence of cancer. But this recurrence can be detected by serial measurement of cancer markers after the treatment is over. Thank you.